Welcome to Life in Biology. I'm Dr. Joel Graff. I'm making a video here for my advanced cell molecular biology and genetics students. This year at Montana Tech, we have taken my research program and brought it into the laboratory, the teaching laboratory. And so the students have been learning to do research based on the research program that I've been building up at Montana Tech over the last several years. So this is going to be about my research, but it's also going to serve as a resource for students in my labs to go back and watch this video, because I present this slideshow at the beginning of each semester, and I'll walk through these slides so they could study it later if they need. Anyway, so in my research lab, we study antiviral proteins, and those antiviral proteins, let's see, it's not wanting to change slide. To understand antiviral proteins, we need to first talk about what happens when a cell gets infected with a virus. So, let me see, I'm going to change this to a pointer, maybe. Oh, it's not going to be a pointer because I'm not in presentation mode. Anyway, when a cell, that's what this is, we've got a cell plasma membrane, the big circle, and the little circle is the nucleus. When a cell is infected with a virus, which is, in my world, it's a hexagon with a little squiggly line in it to represent the genome, the virus goes into the cell, the virus uncoats, and uncoating means that the viral genome is released into the cytoplasm. There are proteins in the cell, like this Pac-Man shaped thing here, that can recognize the receptors for viral molecular patterns. So there's something different about the virus genome than what you'd expect from regular nucleic acid that's in the cell, and these pattern recognition receptors will sound the alarm. I'm leaving out a lot of the steps of the pathways here, but the end result of a pattern recognition receptor being activated is that signaling downstream ends up causing the activation of three families of transcription factors. One is the AP1, another is NF-kappa B, and the third is the interferon regulatory factor family. In this case, I'm pointing out IRF3. When those transcription factors become activated, they will go from the cytoplasm to the cell's nucleus, and the promoter of the interferon beta gene has a bunch of binding sites. It's called the interferon beta enhantiosome, and at this enhantiosome, these transcription factors from these three families will bind, and then they coordinate to upregulate the induction of the interferon beta gene to be transcribed. You transcribe is when you read the DNA and make an mRNA, so we've got interferon beta mRNA out here. Then you have translation, and you get the interferon beta gene being shown here, or interferon beta protein being shown here, excuse me, and through the secretory pathway of the cell, which is not drawn out well in this figure, interferon beta releases from the virus infected cell. It's possible that the interferon could act back on the cell that made it, but in this case I'm drawing a nearby cell, and the interferon goes and acts on an interferon receptor of a neighboring cell. The interferon receptor binds to interferon beta on the extracellular side, but on the intracellular side, the receptor has some proteins from the JAK-type family, and these are kinases that become activated. They phosphorylate each other upon the receptor binding to interferon, and the phosphorylated form is then able to activate some STAT proteins that also interact with another protein called IRF9. This trio of proteins, it can be called the ISGF3 complex, will move to the nucleus, where it can bind to promoter sequences that have a specific sequence called a ISRE, which stands for interferon stimulated response element. So there's a consensus sequence that this transcription factor complex binds to, and there's lots of genes that have this binding site in their promoter. So a huge number of genes, collectively known as interferon stimulated genes, are transcribed and made into interferon-stimulated gene proteins. And these proteins, it depends on the cell type you're dealing with and other factors, but it could be up to 300 different genes are upregulated in response to interferon beta binding to its receptor. So with all of these interferon-stimulated genes being upregulated, the cell coordinates an antiviral response. Because there's quite a few genes here, some have been studied well, but there's still plenty to learn about many of the other genes in this family. So some of the proteins the proteins we study in our lab, some of them at least, are upregulated in response to interferon beta acting on a cell. And one family of proteins we study are called TRIM proteins. The second family of proteins we study are called ring UIM proteins. The TRIM proteins, there's about 80 of them encoded by the human protein, 80 TRIM proteins encoded by the human genome. And the ring UIM protein, there are 4 genes that encode these ring UIM proteins in the human genome. The TRIM protein, TRIM is a shorthand for tripartite motif. We've got a ring domain, a B box, and a coiled domain. Sometimes there's 2 B boxes. Those 3 types of domains are found in the N-terminus of the protein that we study. Meanwhile, with 80 different members of the TRIM family, there is some variability in the family, and so the C-terminus is where there is oftentimes difference from one TRIM family member to the next, and there are subfamilies where you sort TRIM proteins into subfamilies based on the domains you find in the C-terminus of TRIM proteins. Next up, we've got the ring UIM proteins. They also have that ring domain that we had up here, and I'll come back to ring domains in a second. But then we've got a series of zinc finger domains, and at the C-terminus there's a ubiquitin interacting motif. So this protein here is able to bind onto certain types of ubiquitin chains. Ubiquitin is going to be kind of a recurring theme here, and it's because these proteins that we study are E3 ubiquitin ligases. So what does that mean? Well, once a protein is made in a cell, it can be later on it can be modified with different things like methylation, acetylation, phosphorylation, and in our case we study a modification called ubiquitination or ubiquitination, depending on which side of the pond you're on. But anyway, ubiquitins are actually a small protein rather than just a functional group like phosphate, but these small proteins start to get added to the target protein. And when they're added to the target protein, they start to build up a chain of ubiquitins. And if the chain is built properly, it is a signal for this target protein to be degraded. Okay, so not only do cells have a business go about their business making proteins through the central dogma of biology, but once proteins are made, you might have to, or the cell might have a reason to get rid of proteins after a while. So building proteins and breaking down proteins are 2 sides of the story. So how does this ubiquitination happen? Well, ubiquitin is a molecule that's abundant in cells, and it's available as a small protein. E1 is the first enzyme in the ubiquitin enzyme cascade, and what happens is that through ATP hydrolysis, a covalent bond is formed between ubiquitin and the E1 protein. Once that bond is formed, E1 enzymes interact with E2 enzymes, and the ubiquitin is transferred to E2 and is covalently attached to E2. Now there are different types of E3 proteins in a cell. We study ring proteins, and in the case of ring E3 ubiquitin ligases, the E3 protein serves as a bridge by interacting with E2 and the target protein. The E3 brings those 2 proteins in close enough proximity that the ubiquitin can then get transferred onto the target protein, all again through a covalent bond. So this ubiquitin chain here is connected with ubiquitin or with covalent bonds, so it's a nice strong linkage of ubiquitins. We study these E3 with the ring domain, and just a reminder, that was the domain that was consistent between the two families of proteins that we study in my lab. And so we know that the E3s are going to be interacting with E2 enzymes, but one of the goals of my lab is to understand what proteins the E3s interact with that could then be targeted for ubiquitination. I mentioned that ubiquitination can cause a target protein to be degraded, but there's other reasons or other outcomes for when target proteins are ubiquitinated, including changes in activation status of, say, a signal transduction protein. So the overall hypothesis that we go with in my lab can be summed up as guilt by association. So our hypothesis, I'll just read this. If we want to know what TRIM and ring UIM proteins do, it would be beneficial to identify proteins they target. So there are different methods that we are using in my lab to identify protein-protein interactions. So some common interaction assays include assays such as immunoprecipitation and or GST pulldown assays. The second type of assays that you could do are yeast-2 hybrid assays. And for each of these assays, the point of this slide is that you could have two proteins in mind that you want to test whether they interact. So you can test that specific idea, or you could do more of a global search where you're just wanting to know if we have an E3 protein, what are all the proteins it might interact with? And in that case, we'd want an unbiased approach to see just what does bind to our E3 ubiquitin ligase. Yeast-2 hybrid, again, you can have two proteins in mind that you think might interact, and so you can test that specific idea, or what my lab does a lot of are called yeast-2 hybrid screens, and in this case, we, let me close this. We use an unbiased approach where we test whether our E3 can interact with any number, thousands upon thousands of different human protein fragments, and see if they can interact with those. We use a cDNA library for that particular test. But the idea is we can either test a specific idea, or we can go what a lot of people call on a fishing expedition. But here it's not really a fishing expedition because we know these proteins are going to target other proteins for ubiquitination, so I don't really like that term in our use case. So anyway, this is where I stop and I ask my students if they have any questions. I'll move on. So how do we work in my lab? Well, we study different genes of interest, and those would be the 80 different TRIM proteins and the 4 different ring UIM proteins. We order the synthetic constructs of those genes from a company, so we just have to type up the sequence of the gene or copy and paste the sequence of the gene and enter it into a website and hit go, and an instrument synthesizes the gene fragments for us. Once those gene products come in, we have them designed so that they can be cloned into different plasmids. I'm showing 5 different plasmids here. The arrows represent the general flow that we use when we're cloning the gene of interest into different plasmids. The first step of cloning the gene of interest into PGBK T7 pretty much happens all the time. That's almost always our first step. Once we have it in there, we can move it into a kind of a sister plasmid called PGAD T7. From there, we can cut it out and put it into a third plasmid. Meanwhile, from any one of these points, we could do a technique called PCR, which would amplify our gene fragment, and if we design the PCR and the reagents for that PCR correctly, we can design it so that we could then get the gene of interest cloned into a fluorescent plasmid. And we have a number of fluorescent plasmids, and the genes can be popped into and out of those different plasmids so that we can have our gene, our protein of interest that can be tagged with a color. But before I get too much into each of those, the properties of each of these plasmids, I want to go through our general gene cloning logistics. So when we order the genes from the company and they come as synthetic constructs, we have the sequence of our gene of interest plus some extra sequence on the end, at least 15 base pair of sequence that will match up with part of the sequence of this plasmid, PGBK T7. It matches this sequence on the left side of the gene, or on the 5' end of the gene, matches with the sequence to the left of where the plasmid was cut open with restriction enzymes. The sequence to the right of the gene of interest matches up with sequence that is to the right of the cut site when we cut open this plasmid. Plasmids are circular, and we cut the plasmid open by using two different molecular scissors. One's called EchoR1 and another's called BAMH1, which are both located in this MCS, which stands for multiple cloning site. But if we open up this plasmid with EchoR1 and BAMH1 cuts, we can then mix that cut plasmid with the gene of interest with the flanking sequence, and these white X's here are kind of shorthand that mean recombination reaction. And so there's some enzymes that will take the ends of these sequences and the ends of these sequences and cut them back or chew them back a little bit on each one, and then they stick them together. I won't get into the details here, but it's a very simple mix. It's a master mix, and so you just have to mix one reagent with your two fragments of DNA and hope for the best. Next, after you have the gene cloned into that PGBK T7, we can cut open the sister plasmid, PGAD T7, also with EchoR1 and BAMH1. And what we do is, we've got the gene of interest in PGBK T7, we can cut it out with EchoR1 and BAMH1 and then glue it into place using an enzyme reaction called ligation to put it into the PGAD T7 that has been cut open. So this is called subcloning, when you cut a gene of interest out of one plasmid and glue it into the same cut sites on another plasmid. That's called subcloning. And then we can do subcloning again, but this time around there is the BAMH1 site is not in the correct location for cutting out of PGAD T7 and to put into the cut PGEX plasmid that we use, PGEX 4T1. And in this case, we do have an EchoR1 site here, and it's in the right reading frame. Meanwhile, there's an XHO1 site that is to the right of the EchoR1. And if we jump back and take, it's not shown, but there is an XHO1 site to the right of the BAMH1 site in the PGAD T7. So we can cut out the EchoR1 and XHO1 fragment, cut it out here, and paste it into the EchoR1 and XHO1 cut plasmid PGEX 4T1 here. Like I said, at any point in time we could work on cloning our genes into the PEGFP C1 plasmids or other plasmids we have based off of this particular plasmid. And to do this, again, we've got an EchoR1 site and a BAMH1 site. Those are restriction enzymes. They go in and they cut a specific sequence. So there we've got our plasmid has been cut open, which I show again as a C-shaped molecule. And this time around we're PEGFP C1. And in this case, we could take the gene of interest, whether it was the synthetic gene or whether it was cloned into either of any of those first three plasmids, and we can design PCR primers. That's what this indicates here. I have the PCR primer in two colors because the first part of the PCR primer is at least 15 base pairs of sequence that matches up with this sequence to the left of the EchoR1 site on PEGFP, and the red sequence over here matches up with the sequence to the right of the BAMH1 site of PEGFP C1 here. When we do a PCR using these primers that have a yellow part that's a gene-specific part, we get amplification of our gene of interest, and it's a double-stranded RNA or double-stranded DNA construct that not only includes our gene of interest, but those 15 or more base pairs that we included in our PCR primers. So we're at that point. This looks like a lot like with PGBK T7, where we're able to do a recombination and paste or recombine this piece of DNA into this piece of DNA. So that would be recombination. We have other fluorescent plasmids such as M. azurite C1 that is exactly like PEGFP C1, except for some of the sequence within the colored fluorescent protein gene sequence have been changed, so it goes from being a green fluorescent protein to a blue fluorescent protein. But the multiple cloning site and everything else about the plasmids are identical, so we can do that subcloning thing that we talked about a few slides ago, where we can open up PEGFP C1, our M. azurite, cut our gene of interest out of PEGFP C1 and drop it into M. azurite. So why do we do all this cloning? Well, for one, it's great genetic engineering skill development. If students in my lab can clone genes, they'd fit right into any molecular biology lab and have a nice set of skills developed for doing that. But we can also, by putting our gene of interest, our putative antiviral protein gene of interest, into all these different plasmids, we've also created a toolkit to study the antiviral proteins. So now I'm going to walk through these plasmids again, but this time around I'm going to talk about the properties of these plasmids that are useful for analyzing our gene of interest. So again, why all the plasmids? Well, let's dig into that. So when you look at a plasmid, we've seen several plasmid maps already, and I didn't take time to explain them other than the multiple cloning site, which is where you clone your gene of interest in. But a plasmid map is kind of analogous to if you were an electrical engineer and you had a circuit map, or a circuit diagram. So an electrical engineer could look at this and understand how that electric system is going to work, whereas a molecular biologist could look at this and understand what would be the point of putting your gene of interest into this plasmid, and so that's what I'm going to walk through with each of those 5 plasmids that we use in my lab. Okay, so we have PGBK T7 was the first gene that I said that we could put our first plasmid that we could put our gene of interest in, and this is also shorthand. We call this the bait plasmid for yeast 2 hybrid. Why we call it the bait plasmid is because if we have our gene of interest in the multiple cloning site here, and we take this plasmid and we put it into yeast, the yeast can recognize this promoter. This is a promoter from the alcohol dehydrogenase gene of yeast. It's a nice strong promoter, and starting at the transcription start site here, we start making a protein that leads all the way from this first protein sequence on into protein coding sequence on into the protein coding sequence of our gene of interest. So what happens here is that we create a Gal4 DNA binding protein shown in orange. Then there is some extra sequence here between that Gal4 DNA binding domain and where we inserted our gene. That sequence is represented with the white connection line here. And then our gene of interest is cloned into the multiple cloning site. And for our purposes, we clone protein coding sequences that encode the ring domain containing proteins. So we want to use these ring proteins as bait, and how that works is that this Gal4 DNA binding domain will stick to specific DNA sequences and wait around for something to come along and take the bait. Meanwhile, the sister plasmid is not, the first one was called a bait plasmid, this one's called the prey plasmid, PGBK T7. And this prey plasmid looks a lot the same. Here we've got that promoter for alcohol dehydrogenase 1. And then we've got some connecting sequence, and then we've got our multiple cloning site. So when the promoter of one transcription starts here and ends down here, we have the fusion protein encoded, where at first we're making the Gal4 activation domain, which is shown here in green, and we also have some connecting sequence shown in white, and then we also have our prey protein, which was cloned into the multiple cloning site. So these are all made together as one giant protein, but they fold independently with that linker sequence in between. So how does the yeast to hybrid work? Well, our protein of interest, our ring protein, it would be the bait protein here. And remember, we put it in the PGBK T7 plasmid so that it would be stuck to the Gal4 DNA binding domain. Gal4 is a transcription factor. It has two parts to it. It has the DNA binding domain part, and it has the activation domain part. The binding domain is pretty self-explanatory. It binds to specific DNA sequences. But the activation domain, what that does is it recruits in a polymerase. So if we are trying to find out what protein our bait protein is interacting with, we may want to test out a whole bunch of different possibilities for prey proteins. And if there is a nice fit between the prey protein and the bait protein and they interact nicely, we will have the Gal4 DNA binding domain connected to the bait, connected to the prey, connected to the Gal4 activation domain. This is all built up right on that promoter of this particular gene, and so a polymerase would be brought in and it would transcribe the reporter gene. The reporter genes in this case can be determined through experimental tests, whether those reporter genes were turned on or off, or whether they turned on or whether they stayed off. And if they interact, they would be turned on. And in general, the outcome is that the yeast would be able to survive on media lacking certain extra nutrients. Okay, so we're going back to the PGBK T7 plasmid, but this time we're going to talk about it not for use as yeast to hybrid, but for use for immunoprecipitation. This time around, we don't use the yeast promoter because there's another promoter here. If you had eagle eyes, you'd notice there's a P, and then it explained what kind of promoter. Here's a P, and then it explains what kind of promoter. The promoter is T7. It's right in the name of the plasmid. And what this does is it will, if we have a T7 polymerase, the T7 polymerase will bind to this promoter and actively transcribe this gene here. And if there are material around for translation, the mRNAs would be translated into proteins. This can all be done in vitro in a single tube. And so this promoter for the T7 will start transcription here. And I ignored this little triangle last time, but I want to talk about it this time because what this triangle is a MYC epitope tag. Epitope is a term we use in immunology. It's what a T cell receptor or B cell receptor sticks to. B cell receptors at some point turn into antibodies. We won't get into that. But this MYC epitope gives us a few amino acids that can be recognized by a protein or by an antibody that can recognize that MYC sequence. This is great because that means we don't have to get a different antibody for every single different protein we want to study. We can use the MYC antibody or the MYC tag. And so if you express your protein of interest, which in this case is the bait, using the T7 promoter in an in vitro transcription and translation reaction, you will get a bunch of this protein being made. Again, we've got the bait protein, some linker sequence, and then we've got the MYC tag here. And so that MIC tag gives us a little handle, and what we can do is immunoprecipitation relies on using an antibody to grab proteins out of a solution. We can use that MIC tag, we use a MIC antibody to grab this and pull it out of solution. I'll have a better drawing in a couple slides. But let's switch over to talking about the the sister plasmid PGBK T7. Now this one I'm going to change the background color to gray so that we can actually see that I did put a tag on this one as well. So what we have here is the promoter for the yeast, but we're not going to be using it in yeast, so that is irrelevant. This GAL4 activation domain is irrelevant because we're not going to transcribes from here. What is relevant is the part I circled, and that is the promoter of the T7, along with the multiple cloning site where we put in our gene of interest. So if there is a protein that we think our gene of interest interacts with, we will clone it into this multiple cloning site. And we express this also in a T7 in vitro transcription translation reaction. And from that reaction, we will get this particular protein, the prey protein, with a few extra amino acids linking to, this time it's not a MIC tag, but an HA tag. And it's denoted here with a black square, and it says that's an HA epitope tag. So we've got the HA epitope tag on the prey protein and a MIC tag on the other protein, and so we can grab onto either one of these with an antibody that recognizes the MIC or HA tag. We can also detect them in Western blots using the MIC antibody or the HA antibody. Alright, I'm going to switch this one to gray again too, just so that we can maybe... Okay, so we do immunoprecipitations. I wrote out that we have two options for doing an immunoprecipitation, and we probably will want to try it both ways when we're trying to prove that the prey protein and the bait protein interact. Under option 1, we will immunoprecipitate using an anti-MIC tag. That was this white triangle here. So we will grab onto this MIC tag with an antibody, pull this out of solution, which will pull the bait out of solution. If the bait was interacting with anything, it will pull that thing, hopefully the prey protein, out of solution, and then the prey protein has the HA tag with it. So we pull that all out of solution, we run it on a gel and do a Western blot, and we look for the MIC tag to make sure that we pulled down the bait protein, and we look for the HA tag to see if the prey protein came down with the bait protein. Option 2 is just a reverse of that. Instead of pulling this potential complex out of a solution using a MIC tag, we use an anti-HA antibody for our immunoprecipitation. We pull the prey protein out of solution, and if the bait was interacting with the prey, we should be able to detect that on a Western blot by using an anti-MIC tag. We can use anti-HA to make sure we pulled our prey out of solution, and an anti-MIC to see if the particular bait protein in our mixture came along with the prey. A somewhat similar type of assay is called a GST pulldown, but this time we're not making use of antibodies to pull the bait protein out of the solution. Instead, we have some beads that are coated with a particular substance that a protein called GST will stick to. GST is glutathione S transferase. I believe the substance on the surface of the bead is called cephros, and this portion of the GST protein will stick to the beads. Well, if we have those, we can use IPTG, which is a pretend allolactose molecule, and it turns on the expression of this particular turns on transcription from this promoter in bacterial cells. And so if you have this promoter turning on transcription, you make the GST part, which is shown in orange, you make a few extra amino acids shown in white, and then the multiple cloning site on this particular plasmid is right here after this GST protein. And if you clone your gene of interest into this gene, we use the echo R1 and the NOT one, or excuse me, echo R1 and XHO1, and you can see that you go from making the GST protein, you make a short sequence that has a cleavage site if you need it for thrombin, and then it has your gene of interest. So once you express that in bacteria, you can purify the GST bait protein from breaking open the bacteria, and the bait protein can then be mixed with lysates from human cells, for example, or from an in vitro transcription translation reaction, or from any number of other things, and we can test whether the prey protein interacted with the bait protein and comes out of the solution when you spin that bead out of solution. So similar to immunoprecipitation, we just aren't using an antibody, we use a different known interaction to pull our bait out of the system. Okay, and then the last set of plasmids to talk about are those ones that have the fluorescent proteins in them. So here, this particular plasmid has a promoter that has the CMV immediate early promoter. This is a strong promoter that will cause the transcription of this EGFP protein, which stands for enhanced green fluorescent protein. I just wrote GFP. It's a green protein, so I used the green as the color code. Then there's some linker sequence, and then you put your gene of interest into the multiple cloning site. And in our case, we would maybe put our bait protein into the multiple cloning site here. And if we put this plasmid into human cells, our protein of interest will be expressed, and it'll be linked to a fluorescent protein, in this case a green fluorescent protein, and we can see where in a cell our bait protein might want to go. The GFP doesn't necessarily have any particular area in the cell that it prefers to go to, so if our bait protein does have a preferred place in the cell to go, the GFP will come along for the ride because it's connected. So why would we do all of this? Well, the first use is just that this would be a way for us to overexpress our gene of interest, or our protein of interest, in human cells. I'll come back to why we would want to do that later on. I already started talking about fluorescence microscopy. We can see where this protein goes within a cell. That's localization. Co-localization is if we were to have the prey protein also being expressed with maybe a blue fluorescent protein like M. azurite, and we could look to see if the green and the blue go to the same place within a cell, and that's called co-localization, and that would add to our evidence that maybe the bait and the prey protein interact with each other if they go to the same place in the cell. And then of course we could do immunoprecipitation. So just like we were able to do an immunoprecipitation if there was a MYC tag or a HA tag stuck to our bait protein, we could use an antibody that sticks to GFP and would pull the bait out of solution. Okay, so overexpression in human cells, we have multiple reasons for doing that, and those are for functional assays. Those include transcription factor activity, which are luciferase based. We can look for whether overexpressing our protein increases or decreases the amount of autophagy that's induced in response to a certain signal. The cells can go through apoptosis, which is a mechanism of cell death, and we could test whether overexpressing our protein of interest in the human cells makes it more or less likely for cells to die under different situations through apoptosis. So there's all sorts of different things we can do with all these plasmids. So again, why do we do all this? It's going to provide students in the lab with great genetic engineering skills, and we're also building a toolkit so that we can study antiviral proteins. And again, this is where I would ask my students for questions. So if you have a question about this presentation, go ahead and make a comment in this video. Like and subscribe if you want. Talk to you later.